And so it's certainly a pleasure for me to introduce Emma Griffiths. And she has been an active chair of the Working Group on Data Standards. Emma is based at the Simon Fraser University in Canada, and she and a team in this working group have been working tirelessly at responding rapidly to the crisis that we find ourselves in. And she will be giving a presentation on the work that the phage group has been doing on building a metadata standard for SARS-CoV-2 surveillance. Thank you, Emma.
Great. Thanks for that kind introduction, Alan. I'll just get my screen sharing going. Can everybody see my screen?
Yep, I can see it.
Yep. Great, perfect. Okay, so yeah, thanks for that introduction, Alan. As he mentioned, there's a lot of exciting projects that are going on in the data structures work group right now, and Josefina and Marcella will tell you a little bit more about that later on in today's meeting. But as Alan mentioned, in the next 15 minutes or so, I'm going to tell you about some of the work that we've been doing developing a metadata standard to help improve data harmonization and integration for SARS-CoV-2 genomic surveillance. So the first thing I'm going to do is tell you a little bit about the challenges associated with contextual data, or what we call metadata. I'm not going to spend too much time on that because I think a lot of people that are tuning in today are probably very well aware of those challenges. Next, I'm going to tell you what's in the data standard and what would be the advantages to you if you considered using it. I'm going to answer some burning questions that you probably didn't even realize that you had, and then we're going to wrap things up. So when we talk about public health genomics data, we're of course talking about the sequence data, but we're also talking about contextual data. And when I say contextual data, I mean the sample metadata, the lab results, the clinical and epidemiological information, as well as the methods information that help us that are critical for interpreting sequence data and for making informed decisions based on analytical results. And it's really this contextual data in combination with sequence data that lets us do things like characterize lineages and clusters, identify variants with clinical significance, and to correlate genomics trends with outcomes and different risk factors. So in any public health emergency, especially a pandemic, getting the right information to the right people is absolutely critical. And that can involve sharing information between groups within an organization, but it can also mean sharing data with trusted partners outside of your organization or with international agencies or with public repositories. And it's the variability in data structure. And when I say data structure, I mean the different fields, terms, and formats used to encode contextual data. It's the variability in that data structure that's found in local or private databases that often finds its way out into public repositories. And that makes contextual data harder to interpret and more difficult to use. So as I just said, the real challenge in different data structures is that it makes information less interoperable and it makes it more difficult to integrate. And that means you have to spend a lot more time cleaning the data before you can start to use it. And that can take hours, it can take days, it can take even longer. And if, say, you're trying to combine information really quickly to resolve an outbreak, you don't have that kind of time. And so best practices for data management, for data stewardship, and for data structures are all really critical parts of SARS-CoV-2 sequencing and analyses. So you may already be aware that there are existing metadata standards already. And so you may be thinking to yourself, why has this group of people gone to such great lengths to create another one? And that would be a very fair question. But it's precisely for that reason, because there are different public repository requirements, there are different minimal information checklists that exist, but none of them are quite fine-tuned for public health or for SARS-CoV-2. And often they're implemented in an ad hoc manner. Now in phage, we have a lot of folks that are involved in SARS-CoV-2 sequencing initiatives, be they large scale or on a smaller scale. And very quickly, we realized that we needed something more fit for purpose. And so the phage data standard has been developed by experts in public health, in bioinformatics, and in data standards development. And increasingly, the standard and the associated materials are being adopted by different databases and platforms all around the world, including Baobab Limbs that Dominique will tell you a little bit more about very shortly. Okay, so at this point, you're probably like, that's great, but what's actually in the standard? So the phage contextual data standard is made up of a number of different standardized fields and terms to describe a lot of different types of information, including different kinds of accession numbers and identifiers, sample collection and processing, host information, so where the host is from, what the host was, different kinds of exposure, reinfection, and vaccination information, sequencing methods, fields to describe bioinformatics processes and quality control metrics, fields for specifying lineages and variant information, pathogen diagnostic testing details like CT values, and fields to capture provenance and attribution so that data providers are properly acknowledged. Now, all of the fields and terms were sourced from different case report forms, international and national, public repository requirements, existing metadata standards, and extensive literature reviews. We've tried to map all of the elements in our data standard to existing metadata standards, and where possible, we've tried to reuse fields and terms that already exist. Because we're not about reinventing the wheel, and interoperability is one of our prime objectives. You can also find the specification at the link in the bottom left-hand corner of the slide. And feel free to contact us if you want to get that information again later. So in phage, we're not just about developing standards, we're also about developing the tools and the resources to put those standards into practice. And so one of the first tools that we developed was a metadata collection template. And you can sort of see a bit of a snippet of it on the screen here. And you can see there are different fields and they're color coded. So the yellow fields are what we consider to be required. The purple are recommended and the white are optional. And the required fields are prioritized for surveillance. You can also see that a number of fields offer pick lists of standardized vocabulary, and there's also guidance to help structure format for things like dates. Now, you can only see a snippet of the collection template here, but this actually has over 100 fields in it. But the key thing to remember here is that you are in control of your data. You don't have to use the whole thing. You only have to fill in as much of this as you want and as much of it as you need. So it is a spreadsheet. You can just snip out any of the fields that you don't want to use. If you only want to use the required fields, if you only want to use 20 fields, you can custom the template that you use to your needs and ignore the rest. But the key thing to remember here is that everybody that's using the template is picking and choosing the fields that they need from the same pool, and that's what helps to contribute to interoperability. We also have a number of different supporting documentation. So for example, we have a field level reference guide that comes along with the collection template. So you can see at the bottom of the image there, the first tab is the template, the second is the reference guide. The reference guide provides definitions for all of the fields so you know what information we're asking you to put in there. It provides additional guidance for formatting, and we provide examples so that you can actually see what we mean, what we're asking you for. We also have a curation SOP. So this is a set of instructions that tell you how to get started, how to curate your information, and to generally implement the standard. If you are looking to submit data to a public repository like GISAID or the INSDC, and we hope that you will, we've also included step-by-step instructions for how to perform those submissions. These are all found on protocols.io. We're also working on building some instructional videos that will be embedded in those protocols just to make your life easier. Now, you may notice that the submission forms for these different public repositories use different formats in different fields. Don't panic. Our step-by-step instructions tell you how to go from the phage template to the different submission requirements. And the key thing to remember is that while we have little control over the fields in these submission forms, although it has to be said that we're currently working with NCBI to create a SARS-CoV-2 specific biosample form, that by using the phage template, we can help to structure and harmonize the information that goes into those different variable fields. And this is a really critical aspect of being able to clean up and harmonize information in the different repositories. Okay, so now some rapid fire quick facts. We've already discussed this a little bit. Do you have to use the entire collection template? No, you do not. You only have to use the parts that you need, and we've highlighted the most important bits for you in yellow. Is this standard just for human and clinical samples? No, it's for all the samples. So if you're doing environmental sampling, if you are looking at different hosts like bats, pangolins, cats, dogs, cows, whatever, we've got you covered. So it's for all the samples. Do you have to share your contextual data? No. As I said, you have all the control in this situation. You don't have to share a single thing if you don't want to, but you can if you want to, and standardizing and making your information more interoperable will help you to do that. What happens if the pick lists that we offer don't have the terms that you need? This is a great question that we get asked quite often. One of the things you can do is you can get in touch with us, and we'll provide you with the links to do that. Alan already has. The second option is that the curation SOP tells you step by step how to find a standardized term in existing ontologies. So it'll tell you how to go to the ontology lookup service and find a term for yourself. So that sort of empowers you to find the fields and terms that you need as well. Okay, so what can the phage data standard do for you? So basically, it's a one-stop shop for consolidating data from different data streams. So, if you have your methods information in one place, you have your epi data in another place, you have your CT values in another spreadsheet. This is a one-stop shop to bring all of that information together. And that's really critical because that helps to future-proof your contextual data. It's not just bringing it together, but it's putting it together in a standardized way where all of the fields and terms are defined. So that will help you, even if you're not sharing any information, that will help you remember what everything means down the road when you go to use that data again for different analyses and for different purposes. And finally, of course, when everybody's using the same standard, it helps to increase interoperability. So that helps to harmonize and integrate information across different labs and different databases. Okay, so what I've told you, it was the short version. If you're interested in learning more, our standard was featured on the MicroBinfi podcast last year. So you can pop over to that website and you can have a listen to our episode if you're doing the dishes or something. You can also read more by downloading our preprint. We put that out last year. Stay tuned because we're going to be putting out an updated manuscript very shortly because a lot of things have changed since last summer when we put out our first preprint. Okay, so I'd like to especially thank everyone on the data structures team and all of the contributors who provided feedback and input into creating the standard. Without them, none of this would be possible. Thanks to the Bill and Melinda Gates Foundation for the funding that allowed us to kickstart the creation of phage. And very, very special thanks to all of you for tuning in and for listening. And I'd be very happy to take questions. Thanks very much.